can have it. So, I'm very pleased to introduce this edition of EED 2016's last keynote speaker, Ehab Abouheif. Welcome very much. He's professor and EWR Stacey Memorial Fellow at McGill University in Montreal, Canada, and he's currently the past president, if that makes sense, of the Pan American Society for Evolutionary Developmental Biology. He grew up in Montreal in Canada, where he did his undergraduate and master's degrees, and he then moved to the US, and he did his PhD at Duke University in 2002 with Greg Ray. And after that, he was a postdoc fellow with Nipam Patel at Chicago, and then in Berkeley, California. 2004, he went back to Montreal to join the Department of Biology at McGill as assistant professor, and then Canada Research Chair in Evolutionary Developmental Biology. Professor Abouheif works at the intersection of ecology, evolutionary, and developmental biology, which tries to uncover the rules by which genes and environment interact, and how this interaction influences the development and evolution. He uses the complex societies of ants as a model to understand the major phenotypic transitions in ant evolution. So, welcome indeed.
Yeah. It's a real pleasure to be here. By popular demand, I was told that I had to show a picture of my newborn and thank him and his mother for letting me come. She said, "You better give a good talk," so there's no pressure here. But okay, just a couple of announcements and acknowledgements before I start. As Graham had mentioned, I'm the past president of the Pan American Society for Evolutionary Biology, and our first meeting was held at the University of California, Berkeley last year, and our next meeting, the second biennial meeting, is August 19th to 23rd of 2017 at the University of Calgary, and you can get lots of information at evodevopanam.org. Our last meeting, which several of you in this room attended at Berkeley, was quite a success. We had lots of fun. I think the two societies mutually synergize each other. It's fantastic. This meeting was put on by a lot of hard work by people in this room, by the previous council, and the president, now ongoing president, is Karen Sears, who will hopefully lead us into the next meeting and into the future of the Pan American Society. Okay, well, I'd like to thank, of course, the Euro EvoDevo for this invitation. I'm, very much in gratitude for sort of the privilege of being able to give the last keynote address, and just to kind of reflect back on, kind of the great intellectual influence I've had from many people in this room and from the European contingent of Euro EvoDevo, especially Mike Acom, Peter Holland, Gerd Müller, Gunther Wagner, who's kind of pseudo-European now, and of course Paul Brakefield. They've influenced my thinking in many ways and have mentored me over the years, and I'm in great gratitude for that, and for many of my colleagues in this room who have had the best time of my life at these meetings moving forward. Okay, Graham, you can start the clock now, all right? Okay, so I have a chance now to tell you basically about the last 20 years or so of work that I've done on a single genus of ants. So I'm only going to focus on the work we've done on this one single genus. And I guess it's an opportunity to tell you in general how ants have sort of contributed to EvoDevo and how this single genus of ants has shaped our thinking in this way. So I don't have much time to say about this, but this is sort of some major events in the history of thought in EvoDevo on the right over here, and this is sort of a timeline starting with Darwin, going through to Haeckel, the modern synthesis, population genetics, and EvoDevo. And it's just to say that ant research, focusing on EvoDevo, has a rich and complex history that has basically been going on through this entire period. Unfortunately, I don't have time to talk about the contributions of these researchers, just to say that there's a rich and complex history that when you examine it in detail, actually has the opportunity to potentially change the way we think about social evolution, especially post-Hamilton, and also the way we think about the history of EvoDevo. But for now, what I'm going to do in these next 40 minutes is just concentrate on the last 20 years here, and hopefully tell you something about the evolution of complex system in ants. Okay. Many of you are familiar, of course, with ants. I don't have to give too much introduction. They are they belong to a single family that evolved approximately 200 or 150 million years ago. They're incredibly morphologically diverse. They're ecologically dominant, evolutionary, successful. According to E.O. Wilson, the famous myrmecologist at Harvard, that if you were to take all humans and kind of combine them in a single blob, and take all ants and put them in a single blob, that they would be equivalent blobs. Okay, how you would test that, I don't know, but at least that's the sort of comparison with which sort of the scale of evolutionary and ecological success that they've had. And there are two principal reasons for this success. Of course, that they are eusocial, that they've evolved eusociality, and they have remarkable degrees of cooperation. That they, within single colonies headed by a queen and several workers, and the colonies cooperating with each other. And of course, the other reason is that ants are polyphenic. The idea that, of course, like honeybees, when a queen lays an egg, that egg has the potential to either become a queen or a worker, depending on the environmental conditions that egg is brought up in, either by nutrition, temperature, and social interactions. And of course, the phenotypes are quite dramatic. If the egg develops into a queen, the genome now has the capacity to make the queen live for almost 30 years. It develops fully functional wings and can develop millions of eggs. And if it becomes a worker, it only lives for about a few months, has no wings at all, and has reduced reproductive capacity. There's a quite dramatic phenotypic difference that a single genome can produce within a colony. Okay, but this is sort of like the typical, we think of this sort of a single queen and sort of having this type of worker that look roughly the same in the colony as this is kind of the typical sort of ant colony you have. But many of you are not familiar with sort of the progress of ant evolution. So I'm just going to give you a sort of a tour of ant evolution. And I think it will become immediately obvious once I sort of talk about sort of the major transitions in ant evolution, why there's a great opportunity here to understand the origin and elaboration of morphological and social complexity. And of course, I started to write it down, but I think I heard the word complexity at this meeting at least 100 times. Complex structures, complex developmental systems, complex neurobiological structures, and so obviously this is a major theme in EvoDevo. Okay, so we start with the origin of ants, the first major transition towards eusociality, going from a solitary state to a eusocial state. And basically, this is like sort of the origin of eusociality, and all of these have roughly similar sort of character. All of these basal branches, there's about 300 ant genera, and these are sort of occupying the basal part of the tree. And these lineages of ants, these basal lineages, are characterized by very small colonies. Colonies are about 40 to 50 individuals. There's hardly any morphological difference between the queen, which you see on top, and the workers here on the bottom. The only major sort of morphological difference you can see is the difference in that the queens have wings and the workers don't. That's a key sort of difference that happened, is that they evolved this wing polyphenism just here at the early. This is a major morphological difference. And the kind of conflicts that happen between, there's a beautiful behavioral regulation in these colonies. The conflicts that happen in these colonies are quite high in the battle for who's going to reproduce, because it turns out that the workers and the queens have similar reproductive capacities in these colonies. Then there's a second major transition that happens in ant evolution, where now, and E.O. Wilson and Bert Holdover have called this the point of no return. And you can actually draw a line on the tree right here. It's sort of like, it's quite remarkable. And basically, this describes this node, and all the descendants of this node have a similar character. And that is now that the colony sizes explode from now just 40 to 50 individual to thousands of individuals. And you evolve a great distinction between the queens and the workers, as you can see here in this monomorium species. And the workers, typically in most of these lineages, are fairly similar looking. But now conflicts have gone way down, colony sizes have exploded in size, and this is sort of your more typical type of ant colony. And then there's a third major transition that happens in the ants, and sort of the focus of what we happen today, but now it happens only on about 8 times independently, marked by these dots, where now this uniform worker caste now splits into several subcastes. And sort of a typical example of this are the leafcutter ants, where within a single colony you have the big soldiers, the small workers. In fact, in leafcutters there's about 16 different morphological worker castes in this one colony. And this morphological transition has taken the ants now to an extreme evolutionary success and ecological dominance. And you can just see what a single here ant colony of the leafcutter ants looks like. That Bert Holdover, you can see this in its full glory in Ants, Nature's Secret Power. It's a documentary where Bert Holdover actually dug these up with cranes and put 10 tons of cement in the colony, dug it up, and this is what one colony looks like. And this is an aerial photograph by Lawrence Allen Smith in Texas, where you can see the leafcutter ant colonies right here, and you can see the amount of sort of green that it's cleared from the top. So I think just that speaks for its own about the sort of the ecological dominance that these have had. And then the other dots, of course, are the famous marauder ants in Malaysia, which are incredibly dominant. And then, by no introduction, the nomadic army ants, which can easily take down small vertebrates. And here are the soldiers with these big sort of mandibles, and the small miner workers. And the genus that I've been working on for 20 years, here the Phaidoli, which are these are called big-headed ants that have the big-headed soldiers and the small miner workers. And by far, at least in the New World, the most speciose genus in ants. Okay, so we're going to focus today on this third major transition. We're going to try and understand how these multiple subcastes came about. And this is not something that occupies me. This has gone back to occupy some of our most famous biologists over time, including Wilson, Julian Huxley, and others. But here's Darwin on the subject. He spends quite a bit of pages in the origin of species. And Darwin, in fact, had 2 problems with ants. The first was that the workers were sterile. And if natural selection is working on the level of the individual, how do you find sterile workers like within the colony? But that's not our subject today, and that's something that Hamilton, and that's something that has happened of great debate in the pages of Nature recently. But he had a second major concern, and that's written over here. And he sort of, after describing the first concern, he says, but we have not yet as touched on the climax of the difficulty, namely the fact that neuters of several ants differ, not only from the fertile males and females, but from each other, sometimes almost to an incredible degree, and thus are divided into 2 or even 3 castes. Okay? And so Darwin spent some time trying to figure out how this could happen. He presented some scenarios where he thought that, okay, well, maybe he spent some time looking for morphological characteristics that would connect these different subcastes together. He envisioned that you had a sort of continuous distribution of workers that all were connected somehow morphologically, that slowly and gradually, with small amounts of selection, would split and then develop into these 2 things. And if that was the case, you should find things that would morphologically link these 2 subcastes together. But he wasn't very successful at finding them. Came DeWitz, or Dewitz, I should say, sorry, in 1878, a German biologist, who was in fact the first one to describe imaginal discs in any insect. So this is quite an achievement and part of the history of social insects and developmental biology. And he responded to Darwin. And his sort of major contribution was that let me read what he says here. He says, in his Origin of Species, Darwin speaks of the difficulty imposed onto his theory by the so-called neuters of eusocial insects. in particular worker ants with a morphology differing greatly from reproductive animals, caused by the complete lack of wings. This prompted me to investigate whether at least rudiments of these limbs, which are encountered so often in orthopterans, can be found. For a long time, my search was in vain until I examined larvae and found imaginal discs at the size of the two back thoracic rings. And so basically what Dewitz was trying to say is that instead of looking for morphology, links between these subcasts by morphology, he tried to look at development. And so he has these beautiful pictorial diagrams. This is an ant larvae, the head is over here, and these two are, these are worker larvae, and so there's not a single case where workers and ants of any species have wings. The workers are always wingless in any of the 15,000 species of ants. Yet, when you look at the larvae, you find these basically vestiges of wing imaginal discs, which are precursors of the adult wings. And so here are the three basically imaginal discs that will develop in the adult into the leg discs, and here are the vestigial wing discs over here in 1878. And basically, since 1878, it was another about 100 years before we realized that, like I said, these vestigial discs are actually a universal feature of all the ants. So you find two things. The wing polyphenism, the ability to produce wings in the queens but not in the workers, is a universal feature of ants, of the 15,000 species. And when you look at larvae in their last developmental stage, you see the appearance, the transient appearance of these wing vestiges that appear in the larvae. So that brings me now another 20 years, basically to 2002, when I was starting my PhD, and wanting to ask basically, well, how does this happen? If these are universal features of ants, how is this wing polyphenism regulated at the developmental and genetic level? And of course, at that point, I was able to build on the sort of the brilliant work of developmental geneticists that had worked out this sort of wing regulatory network in Drosophila to some detail. And so here it is, basically, without going into too much detail, it's just familiar to many people, but of course, many signaling common, very familiar signaling pathway, signaling molecules, transcription factors, working in this quite elaborate way to control patterning growth and differentiation of Drosophila wing imaginal discs. And just to say that, in general, when you look at the winged queens, this network is quite conserved, highly conserved. So this is the wing imaginal discs of Drosophila, and this is a gene called scalloped. You can just go back. Scalloped is right here, SD. And if you look, just to give you an example of the conservation of this network, you look at the expression. It's expressed in the future wing pouch of Drosophila, and here it is, the future wing pouch of ant queens. And if you just look at the expression, actually real expression of scalloped, here's what it looks like. And if you look at any of the genes, over the last 20 years, we've looked at most of the genes in the network in these wings, and the expression patterns are remarkably similar, sort of implying that these networks are conserved. And so in 2002, I basically published a paper in Science showing that, well, the Drosophila network here is with most of the genes in green, of course. Much of this network is conserved in butterfly wings, and here you have the ants with the origin of the wing polyphenism. And when you look at those, the casts that produce wings, either males or the queens, whenever we looked at these genes, you see that these networks are conserved. And of course, again, I've added to this quite substantially over the last several years. But the point is that we had predicted that, well, just going back, we had predicted that since wing polyphenism was a universal feature of ants, and they all had these vestigial wings, that you would find in the wingless casts in all of these species, that you would interrupt the network in similar ways in all of the species. I mean, evolution is quite lazy. These networks are highly conserved. So you would think that you would find a single place that you would interrupt these networks. But when I looked, okay, one of the big surprises is that I found that in different species, you interrupted the networks in different places in the different species. So while you had a conserved network in the winged cast, in the wingless cast, you were stopping this network, you were repressing certain genes in this network in different places in the different species. And so this was quite a surprise. And I have to say, I've spent, seriously, since my PhD in 2002, I have looked to try and, I've looked hard to try and understand why this is the case. And we've come up with different models to say, well, is it natural selection that's driving these network to be stopped in different places? Is it neutral drift? Is it just that, you have to get rid of the wing anyway, so these networks are allowed to drift neutrally? Is it some combination of neutral selection and drift, natural selection and drift? But one thing I didn't do, and this takes me back since I'm here in Euro, and I don't know, Michael, are you here somewhere, Michael Acom? Or did you, oh, he went home. Come on. This is like great, he gets a chance to like tell me I was right, after all these years. I went to his lab. I went to his lab for a postdoc interview. I interviewed at Cambridge. And when I presented this, this part of my PhD, he just said, I sort of said, well, it could be neutral drift, it could be natural selection. He said, Ehab, I would be very careful about saying that this would be neutral. And, those words didn't quite fall on my shoulders right away. And so I kept looking for signatures of selection in the genes. I kept looking at the network, and I looked at expression patterns, trying to knock out the genes, and let's do CRISPR, and let's do RNAi, and let's do all of this. But one thing I forgot to ask is, what the hell do these vestigial wing discs actually do? What is their function, right? And this is, of course, the blinding effect of always trying to look for the gene, the genetic basis, and forgetting about the actual, some of the development. 20 years. And anyway, so finally in the last few years, we decided to find out. The assumption was, you can't necessarily blame me too much, because when you think about transient vestigial structures, the classical textbook case, look at any textbook about vestigial structures, they'll say, yeah, these vestiges are good. They're evidence of evolution. Darwin used them extensively to talk about the evidence for common ancestry. Our current textbooks talk about the evidence of ancestry. This goes to the, the hind vestigial structures, the pelvic bones, the hind limbs in whales, or the little tail, the existing tail in the coccyx. There it is. The coccyx in humans, the little tail. And so, they'll tell you the evidence of ancestry, but we don't think of them as necessarily having functions or any major functions in evolution. And so I went on thinking, oh, they probably had a minor functional role at best, but nothing major to it. So finally, we decided to try and test what the function of these is, and this is work that was led by Raja Kumar in my lab. He's a PhD student. He's now a postdoc in Marty Cohen's lab. He's on the job market, remarkably talented individual. And he's led this project with several other of my grad students in the lab. And so we set out to do this work with one species called Phydolia hyetidae from Arizona. We found it. And of course, it has the typical minor worker and the major worker here that you see. Okay, and if you look at a typical colony of Phydolia hyetidae, they have often 1 to 6 queens. The soldiers are found about 5% of the colony, the minor workers about 95%. And we know something quite a bit about the caste determination switches in Phydolia. When a queen lays an egg, this is a sort of a temperature-dependent switch. If it's springtime, the temperature is getting higher, the queen releases juvenile hormone in her abdomen, the eggs that she lays become queens, they develop into these large larvae, they produce 2 wing precursors, the 2 wing imaginal discs, and they become queens, virgin queens with fully functional wings. They fly off to find mates. But most of the year, in fact, the eggs that the queen lays have low levels of juvenile hormone, and they turn into workers. And in the last larval instar, this is a nutritional switch. Just depending on the amount of nutrition that the larvae are fed, you'll either become a soldier or a minor worker. And if you're a soldier, you develop into a larger larvae than the minor worker. You have development is delayed about 9 days. And you develop, curiously, this vestigial 4-wing disc that develops in the soldier, turn into normal soldier that's completely wingless, even though it has this vestige. Or a minor worker, there's no visible wing disc in the minor worker, and then into the sort of the minor workers here. And just to think about, it's kind of curious that when you look at this vestigial disc, if you look at the growth of wing discs in the queen, that they start to grow very early in larval development from the very first larval instar. They grow, and that's by the green over here, and they grow into fully mature discs starting very early. But over here, this disc doesn't grow, this soldier forewing disc, which is one of the major differences between minor workers and soldiers. It doesn't grow until you get this juvenile hormone pulse turning the larvae into a soldier. Then this disc grows, and it starts growing at an incredibly fast pace, even faster than the wing discs in the queen. And so you ask the question, okay, well, you know, this disc is growing incredibly fast, and then at the end of larval development, there's a very coordinated shutdown of this disc. So if you're a queen, sort of near the end, you're a well-patterned disc. This is the expression of spalt. You have it in the sort of the wing hinge with the spalt stripe in the middle, just like in Drosophila. And this is the expression of apoptosis, cell death. There's really no cell death, nothing to write home about. But if you're a soldier, near the end here, after this incredibly fast burst of growth, here you get the expression of spalt in the sort of the wing hinge, but it's down-regulated in the pouch. This is apoptosis, not really any apoptosis going on. But then as soon as you go from the larval to prepupal transition, spalt activates in the middle, and in those same regions that spalt is, you get apoptosis, which then spreads and kind of chews up the disc, and it shuts down. So for a vestigial structure, there's quite a coordinated burst of growth of this thing. Again, if we just go back, it's like there's quite a coordinated burst of growth right from here, and then near the end, this thing shuts down by apoptosis. And why would we, you know, it's kind of curious, why would this happen? Why would a vestigial structure have such a coordinated sort of burst of growth and then a shutdown by apoptosis? So what we did is we decided to try and, we wanted to knock down this gene. This is a gene called, right here, Vestigial, which is basically a sort of a wing identity gene. This is the work of Sean Carroll, Garcia Vito, and many others, that controls basically the growth, patterning, and differentiation. If you have vestigial expressed, it means you're going to become a wing, at least at the larval stage. And it's expressed just as it is in Drosophila, sort of at low levels in the pouch and at high levels around the dorsal ventral margins of the disc in the queens. And at this stage in Drosophila, it's wing specific. It's only expressed in the wings. And it's the same in ants. Here's again vestigial in the wing disc. This is the head. There's no expression in the head regions. And these are the 3 leg discs. You see no expression in the leg discs. It's really something that's expressed exclusively in the wings. And then over here, you see this is the soldier. That's that soldier forewing disc that I was talking about. You see, the expression of vestigial is conserved. It's not in the head or in the wing discs at all, just as it is in the reproductives. So what we did is we then proceeded to use RNAi against vestigial. Right here, as soon as the thing became a soldier, we injected RNAi into the larvae to try and knock down these discs. And we were successful. We were able to, this is sort of injecting a YFP control. This is sort of normal. Here are the vestigial wing discs on this side. And these are the vestigial RNAi discs. We're able to reduce the growth of these vestigial discs successfully. And to check the phenotypic effect of these, to sort of look for the phenotype, we looked at the pupae. Basically, you can see this is a minor worker pupae that will turn into a minor worker, and this is a soldier pupae. And basically, after this, as soon as they become pupae, there's no growth that happens. They basically come. It's just easier to look at the pupae. And here, after 20 years, is where my jaw dropped when I saw this result, when Raji came knocking on my door. Here's the control minor worker, and here's the control soldier. When you knock down the wing disc, you not only made the soldier smaller, but you actually reduced the head size of the soldier. So it took me a little while, but it means that this little vestige, or this wing disc, has now been co-opted in ant evolution to control head and body size regulation. It's kind of must be sending signals to the head and the body size to control sort of the allometry of this. And so, people, here's the sort of, if you plot the variation within a single colony, head width versus body length of the minor workers and the soldiers here along this line, I've tried myself in my own lab to play with juvenile hormone, and you can never get anything sort of in between. You can either get bigger minors, smaller minors, bigger soldiers, smaller soldiers, but nothing in between. But when you knock down with vestigial RNAi, these are the red dots here, you can basically connect the minor workers and soldiers in terms of the bodies. And here are some examples of adults that come out. Here's again the minor workers and the soldiers. You can produce a whole range of intermediates that basically, because RNAi is a knockdown, it's not fully penetrant, so you get a range of effects. And there you can see some things that look a bit more like minor workers, some things look like matrix. Okay, so what does this all mean? It means that you basically, if we were to put the whole scenario together, you have social interactions that basically you feed certain larvae nutrition that increases JH, it induces the growth of this disc, and then this disc goes to control head and body size and allometric scaling in the ants. And so this is kind of quite remarkable. But what happens in nature is the big question. So, I mean, okay, so you mess with this thing in nature, but what happens, what's sort of the major dynamic of these things in nature? This is sort of the eco part of the eco-EvoDevo. We wanted to find out. And so here you have basically this notion that they control this ratio of 95% minor workers, like I told you, and 5% soldiers, right? And there's this complicated social interaction that keeps these ratios the same. And it was discovered early, this was basically in the 1940s, and later sort of elucidated in the 1980s by Diana Wheeler, and this was the work by Greg, that there's actually a soldier inhibitory pheromone that helps keep this regulation right in place. So if there are too many soldiers, the larvae lick this or they contact the pheromone with the larvae, and they can stop a soldier from becoming a soldier. And this is basically some very elegant experiments by Pasteur in 1974. Here you have basically the number of soldiers in a colony on day 1. You either have a colony with 0% soldiers, 5%, which is the normal amount, 10, 20, 50, or 100. And then you look at the number of soldiers after 75 days. So basically, this is the kind of what you would expect, 5%. Regardless of the number of soldiers, this soldier inhibitory pheromone should keep them around the 5% mark. If there was no inhibition at all, you would, after, if you have 10% on day 1, after 75 days, you would have 10%, and then so on and so if there was no inhibition. And basically, when you do the experiment, it's quite clear. This is the red line. On day 1, if you start with 10%, after 75 days, it knocks it down to 75. It's even more amazing. If you start with day 1 with 100% soldiers, after 75 days, you even have less than 5%. So there's a really strong inhibition. The soldiers are really detecting if there's too many soldiers, and they inhibit them with this soldier inhibitory pheromone. And so there's this real dynamic of activating soldiers through the wing disc and juvenile hormone, and then inhibiting them. And so what we wanted to do is we wanted to see what the mechanism is of this soldier inhibitory pheromone. So what we did is just like the vestigial RNAi, right here, just after juvenile hormone, we took larvae out and we exposed them to 100% soldiers. And we wanted to see what happened. And basically, you get the same kind of result that you do when you knock down the disc. You basically, here's the control worker, here's 100%. You reduce the size of the larvae and the head width of the disc. And when you actually look at the discs in this experiment, the soldier inhibitory pheromone is doing so by actually inhibiting the growth of this disc. So the ants can activate the growth of this disc through nutrition and juvenile hormone, get the thing going, make the soldier through this wing disc, which is signaling to the head and the body. But they can also lick this inhibitory pheromone, which is going through the larvae and inhibiting the disc so that it stops its growth. Okay? And if you look, you get the same kind of spread. This is the minor workers and the wild type soldiers. You again can produce all of these intermediates. And these plots, if you look at the vestigial RNAi on top and this soldier inhibitory, the effect of the oligometerium, there's quite a remarkable similarity between the two. So here, what we're discovering then is that you have this nutrition working through juvenile hormone, activates the disc, makes the allometric scaling. If you produce too many soldiers, the soldiers are going to talk to each other. They lick this inhibitory pheromone on the larvae, and they inhibit this disc. And then you have this complicated system, sort of complex system that's starting to happen. And unfortunately, I can't tell you about these other experiments. We did another slew of experiments that actually shows how juvenile hormone can work either through the disc or independently of the disc to work on body size. And also the inhibitory pheromone can work either through the disc or independently of the disc. And so now we can sort of take a breath and figure out, okay, so what does this actually mean, okay? It means, okay, at this point, okay, that we've been able to unify this kind of superorganismic regulation. If you think about the colony as a superorganism, what this disc does is actually it unifies the organism and the superorganism. It means you're regulating the colony through a property of the individual. And this allows you to connect everything from the gene all the way to social interactions and even probably even, you know, I mean, there's all kinds of, the shift in the ratio of workers, soldiers, this 95-5%, it changes when when the colony is attacked by fire ants or so on. You can arrange this. You can regulate both at the superorganismal level and at the organismal level. So we can really connect now. We have the opportunity to connect the gene all the way to the colony. Okay. The other thing is I just want to make a point that while this sort of system is working quite well at this level, you can see that perturbations to this system can produce all kinds of interesting variation, which is kind of cool. Okay. I just want to say that, you know, you might be asking, well, how did this disc get co-opted to do this function in the first place? And I just want to say something about the amazing biology from the 1930s and 40s in Drosophila, wing imaginal disc, going back to Hadorn and others, and two groundbreaking papers in Science that essentially showed that there's a communication between the discs and the whole body of the ants, like a very ancient mechanism. So that here in a wild type situation, you're fine, but there's really no signals that are sent to the body. But if you damage the disc, this disc will regenerate. And in the meantime, while it regenerates, it releases these kind of Drosophila insulin-like peptides to tell the body, it releases ecdysone, and it tells the body, hey, stop. Keep, you know, you should just stop growing until we regenerate the disc. And also, if you have an overgrown disc, if the disc overgrows, you release DILP8, you stop the whole thing, you release ecdysone, and then it sort of knows when to stop growing. So there's this ancient system of communication between imaginal discs and the body size. And what we think is that this system was kind of tinkered with to then use this to actually, you know, send signals to the head and the body size. But, you know, this has really opened up kind of a new, you know, we have to really start thinking about, you know, what this means now and what are the mechanisms and all of these things. The other sort of major implication is, which is kind of neat for complex systems, it means that developmental changes key for the origin of a complex system are critical for elaboration. It means that at the beginning remember I told you at the beginning that wing polymorphism was a major key factor in the origin of ants. And it was just about reducing the wings. And so at that point, you know, there's hardly any difference between the queens and the workers. It was all about getting rid of the wings. At that point, it's all about changing, you know, that's sort of the origin of the complex system. And then as ants started to elaborate and diversify, you can use those same structures that were key to now elaborate the system. And I want to show you just quickly, I don't know how much time I have left. Graham, how much time do I have left? I'll take that as a good sign or a bad sign. He's either enthralled in my talk or he fell asleep. What's that? 3 minutes. Okay. All right. Well, I'm just going to tell you that, you know, now that you have this system in place, it can serve as a major driver of novel casts. So, and I'm just going to try and tell you very quickly, okay, how now this, you know, once you have this system in place, how you can now diversify to produce novel elements in these complex systems. And I'm just going to show you, this is on 8 species, in 8 of the 1,000 Phytoli species, not only do you have minor workers and soldiers, but you also produce this super soldier caste that is in 8 species which all that live in Arizona. And these super soldiers, at least in some species, we know their function. They serve to protect against the army ants. You can see here an army ant sort of attacking a super soldier. What they do is they use their large heads to block the nest entrance. And then what they do is once the army ants walk over this, right, they kind of disband, they attack the army ants, and then they come back like a Spartan shield, and then they block the nest entrance again. And so the idea is, well, okay, so how did you get these super soldiers? It's pretty amazing. When you look at their wing imaginal disc, here you have a soldier, the larvae, that single vestigial disc that I was showing you for Phytoli. Now in this species that has naturally evolved a super soldier, you see that they have two massive vestigial discs. So, you know, I often get the question, okay, big shot, you made a soldier and a super soldier, but can you make a super soldier? Who knows, okay? We'll see what happens. But one of the things is that we thought that since they all belong, they all live in Arizona, that there would be a single origin of these super soldiers. But when you look across the tree of life of Phytoli, this one single genus, you see that in fact, super soldiers evolved, well, actually this is not working good, but the super soldiers evolved twice independently. There's a parallel evolution of these super soldiers. And this parallel evolution occurred through increasing the number and the amount of discs that they have. But, you know, just to say, well, what is the mode of sort of action of this? Well, one day in this one species here, in Phytoli morsi, which occurs right here, this occurs in Long Island, nowhere, no army ants, nowhere to be seen in sort of, you know, no army ants in sight, no super soldiers in sight, okay? One day when Raji and I were collecting these, here's a normal soldier, we found this really large sort of super soldier-like species, individuals in this colony. And it took us actually quite a while to recognize this. We're like, Raji, did you see that super soldier? No, did you see that? No. And then we would go back, and we're driving back to Montreal. Those individuals really large. Yeah, they're really large. You know, we're going back and forth. And it took us a while until we went back to Wilson's book, which he had published. He took 17 years of his life to publish this whole tome on this genus Phytoli. We went back to figure out that, oh my God, that looks like a really evolved super soldier. Except the fact that these anomalies in Long Island that we found, this is like a normal soldier, these were not normal individuals. It was clearly some kind of induced variation or anomalous variation. And what's interesting is you can see that it looked like a super soldier, except it had these two little wing buds on the side. And so somehow you were, in this one population, this natural variation, you activated the super soldier program through the discs, but you also activated the ancient function of these wing discs and produced these little wing buds. And, okay, here we're going, this is good, wait. So now, here, what it means is that we induced in a species that doesn't normally have super soldiers, we're able to induce sort of super soldiers in species that didn't have them, and this is sort of what that looks like in Phytoli morsi. So here's a normal soldier, here's a super soldier, and the ones that we did in the lab, okay, well let me just tell you how we did this. So let me just say, okay, this here we found in Phytoli, this is where we found the natural ones in sort of natural variation. We went back to the lab, and if you hit them with a really high dose of hormone, just before that minor worker soldier split, you can actually then induce the super soldiers. I'm about to show you here. This is what it looks like. And the ones that you induce in the lab with these really high doses of hormone also have these wing discs that you see. So whatever happened in nature was similar to what happened in the lab, okay? And it could have either been that you modified through mutation the sensitivity to the thing, or in nature somehow there was a jack-up of the juvenile. We don't know. The jack-up of the juvenile hormone, we don't know. But we just know that whenever we do this, and we went to any of the species on this tree, any of the 1,000 species, and if you just touch the thing with these high doses of hormone before the minor worker super soldier split, you can induce these super soldiers to appear, okay? So what does that mean? It means that basically the potential here for the super soldiers was actually there. The super soldier potential and expression of the super soldier was there in the ancestor. That you lost the phenotypic expression of super soldiers, but you retained the developmental expression. And we know now, at the time when I published this paper in Science in 2012, Raji and I, it was lost, but we didn't really know the mechanism. Now, with understanding that the disc is what's actually controlling head and body size, we know that that was retained because you have to make a soldier with a single disc. So you can lose a super soldier, but you will always have the mechanism in place to produce a super soldier. And so that retained that potential for over 65 million years. And then that potential, I mean, it's just that basically what it means is that potential is being reactivated randomly in all of these colonies and all of these species around the world. And on occasion, when the conditions are right, you will then regain, you will fix that expression through a process called genetic accommodation to produce then to evolve a natural super soldier. And so I just want you to think about something. I'm done. I just want you to think about something here, is that, you know, Haeckel had basically described this pattern where, you know, you had, you know, ontogeny recapitulates phylogeny. And what he used that pattern for is that all of these vestiges were transiently coming up in development and disappearing, transiently coming up and disappearing, and he used that transient appearance and disappearance to reconstruct animal phylogeny, okay? But what I'm proposing now, it means that basically, and Haeckel's description of that was absolutely general. I mean, he was able to do animal phylogeny because all across the vertebrates and invertebrates. And so all our bodies, all organisms are all continuously, you know, have these vestiges. And we might have thought that they could serve some minor role. And what I'm proposing here, based on the ants, is that those vestiges are probably playing some major signaling roles throughout the body. And they retain these ancestral potentials that can be activated by environmental stress or mutations in the apparatus that sense environment, so that you can induce these ancestral potentials. And that is raw variation for evolution that can be fixed. And that's something that's a sort of a mechanism that hasn't really been sort of appreciated. And it's absolutely widespread. I don't have time to show you the pictures of humans with tails and hair and all these crazy things, but I just wanted to show you Darwin's quote, because Darwin himself, not in The Origin of Species, but in another book that I had never read until recently, which is The Variation of Animals and Plants Under Domestication, Darwin had recognized this. So he posed the conundrum of all of these different castes and how do you explain it? And he answered himself in another book, okay? And basically he says, and it's very humbling, and look at, okay, for all of those who were writing fast to like publish papers and stuff, I mean, this is really inspirational. The dude wrote like, you know, how many books? And he was able to write it so poetically. And here it is. He says, but on the doctrine of reversion, as given in this chapter, the germ becomes a far more marvelous object. For, besides the visible changes to which it is subjected, we must believe that it is crowded with invisible characters, okay? And to a long line of male and female ancestors separated by hundreds or even thousands of generations from the present time. And these characters, listen to this, and these characters, like those written on paper with invisible ink, it's making me cry here, all lie ready to be evolved under certain known and unknown conditions. Okay, thank you very much. These are my students and funding agencies.
Thanks very much indeed for a fantastic talk. Are there any questions? The correct answer is no, but we have got to. Yes. Oh, go ahead. Go ahead. You, yes.
Thanks, Ehab. That was a great talk. In Drosophila, we know that vestigial is definitely expressed in and critical for wing disc development, but it's also expressed in and critical for nervous system and muscle development in both the thorax and the abdomen. And the brain produces all of these hormones that are going to tell. So are we able at this stage to rule out the alternative explanation that the disc, the vestigial disc, is not communicating with the body growth systems, but that the other roles of vestigial in the ant are causing these morphological differentiation? How far can we go with that?
So that's a good question. Basically, the way it goes is that vestigial in ants and in Drosophila, both, vestigial at the early embryo stage is expressed in a number of tissues, right? It's expressed in the nervous system, in all over the embryo. But when it gets to the larval stage, it only then gets restricted only to the wing discs. So we checked this quite extensively because we wanted to make sure that we were ruling out that it could be expressed somewhere else. And so when we're knocking it down at the larval stage, we're exclusively knocking the wing disc. Yeah.
Because the nervous system is one of the few embryonically established organ types that lasts through metamorphosis, perhaps we can't rule out the possibility that vestigial action in the nervous system at early stages or even during larval life could be affecting systemic factors. What do you think?
Yeah, I mean, I think, okay, so, is the disc communicating directly with the nervous, the head discs? I mean, this is now, it's opening up a whole new opportunity for future studies. Graduate students should come to my lab to try and attack these things. Right now, I mean, and then of course the wonderful experiment that's going to take years to do is to actually then transplant the discs from one cast to another. It's going to take a long time, a lot of effort. But right for now, what we can say is that the disc is necessary, not necessarily sufficient, but it's necessary for the soldier, the development of soldiers and regulation of the head and the body. Whether it's the exact, it's connecting directly to the head, that's going to take a few years to figure out.
Yeah. I'll take one more question off here. I'll take this down. No. How you get it? No, wait, this goes first.
Thank you. Okay, so we have long growing lists of examples where losses of complex traits developmentally really are gains of inhibitory functions, right? And we lift these inhibitions and the T1 segment produces a wing and the legless abdomen of insects gains its legs. And do you see your story in essentially vestigial organs in that same tradition, that these more complete losses on the surface still retain that same potential you see here for reawakening structures that then, say, can become claspers in sepsid flies or something like that?
Yeah, I think that's a really good point you make, and I really do think so. I think losses can also be, as long as, you know, and we know that, I mean, one general feature of development is that developmental genes play roles throughout different tissues. And so even though you might lose necessarily the tissue, it could be then expressed transiently with genes that are then kept in other places. So you might never lose it entirely, right? And so I absolutely agree with that. I think that's a nice way of summarizing that, yeah.
We're going up in the balcony.
Just toss, just see what you can do, yeah.
Last question, I'm afraid. We have to move on. Hurry up.
Okay, thanks. Really very inspiring, but I want to go back to the previous question. So it appeared to me, but maybe I got it wrong, but it appeared to me that the only direct evidence for direct involvement of the disc in regulating the growth is that injury experiment, right? But in that injury where you cut the disc, or when you transplant a disc, then you bring into play all the regenerative response, which requires resources, and all that can actually come on the expense of growth, right? So in this case, you really need to be aware and control for that.
So, I mean, that's a really good question. I mean, one thing that we have done is we made sure to measure all the other discs when we did this. And the other discs are kind of unaffected. So in those injury experiments, I mean, it's known, Armin has done these famous experiments in the beetles where, you know, he injures the horn disc and the genitals get bigger, and you mess with the genital disc and the horns get bigger. And so traditionally there's kind of this sort of communication between the discs. But it's a very interesting thing. It's kind of contra what we're finding, because normally when you reduce a disc, okay, like with the communication, like in Drosophila with these experiments, you reduce the wing disc, you make the other disc bigger. What's happening here is that the wing disc is getting bigger and it's making other things bigger, right? So there's not the same relationship. It's really sort of, it's tinkered with it in a way that is making it sort of, it's not just sort of this system that's in place and you just perturb it and it's making these things. I think it's a real adaptive evolution of the disc, because now it's not exactly what you're seeing before. It's now the disc is getting bigger, the other ones are getting bigger. So it's a completely new kind of relationship that has evolved here between communication of the discs. It's not the kind of homeostatic thing that's there where you make something smaller, you make something bigger. So there's a difference between homeostasis, and that's been argued by sort of Alex Shingleton, who's shown this sort of homeostatic and Fred Nyhout relationship. This is really something new and adaptive that's producing, it's really the proliferation. If you knock out the disc too late, you don't get an effect. These cells have to be proliferating for you to get this effect.
Great, thank you very much indeed once more. That's really fantastic. That fantastic talk concludes the business, apart from the business meeting, of the conference. Thank you all very much for coming. Just a couple of things. Some people have bought t-shirts, they haven't picked them up still. This is crazy. They're sitting on the desk up there. Please go and get them now. I'd just like to say a couple of thanks, especially to the technical support we've had, including Peo sitting up in the, stand up please, for technical support. Thank you very much indeed. We've been pretty incompetent, they've really helped us out. Finally, all the yellow helpers, please stand up. Not the DK one in here, but all the others, please stand up. You've done it. You've done a really fantastic job. It wouldn't have worked without you. Thank you very much indeed. Gert will say something now, and that'll be it. If the helpers, if you want to get in here, just come and see me.
Well, as Graham has indicated, we seem to have come to the end of this meeting, unfortunately, because really I enjoyed it a lot, and it culminated in Ehab's, like, don't we all want to go home and work on ants now? It's fantastic. It was a pleasure all over. The executive committee has received lots of very positive feedback. I'm not sure whether it's merely positive because the negative one doesn't filter through, but it was altogether a joy to be here, and that's greatly due to the local organizers, whom we should thank at this stage. We have actually, the executive committee has gotten presents for the local organizers, but the presents are so heavy that they can't be brought on stage. So we have hired special transport to be transported to the banquet tonight, and so the presents are going to be handed over at the banquet. But at this stage, I think we should give the local organizing team a great applause. Now, there have been many more people working behind the scenes, especially several committees. There is the executive committee, there is the scientific committee, there is the program committee, and there is the council and several other parts of the society that have been active in preparing the meeting. And I'd very sincerely like to thank all these people who have been active to prepare this meeting. Thank you. Now, last but not least, because we still have the business meeting and the closing, the banquet tonight, but there is one more thing, and that is the venue of the next meeting, EED 2018. And there has been fierce competition, of course, for organizing the next meeting. And I draw out my virtual envelope, you know, and I pull out and I say, and the winner is Galway, Ireland. I like to ask Gerhard Schlosser, who will be a local organizer at Galway, to briefly introduce the venue, and we invite you all, those who are still here and those who have already left, and hopefully many new ones, to attend that meeting at Galway. And while they're preparing the computer, I would like to remind you that through your registration at the meeting, you're also members of the society until the next venue. So for these 2 years, you're members. And don't forget that. I think the community should be enforced by this fact that even in between meetings, you're members of the society. We have a new homepage up, which is still going to be improved a little bit, but it should inform you about interesting events in between the meetings also. And so I ask you to keep in touch, and I hand over to Gerhard.
Okay, well, thank you very much. And also thanks again for the local organizers of this meeting for an excellent meeting. We like to invite you for the next meeting to Galway, Ireland. We, that's my colleague Uri Frank and myself, who have offered to organize the meeting. I'm sure the moment will come when I regret this, that is when all the work comes close, with still 2 years to go. But I hope you all will be coming. And for those of you who don't know Galway, Galway is on the west coast of Ireland. It's a small town of about 70 to 80,000 people, widely considered the cultural heart of Ireland. So there's a very lively pub scene. It's less famous for its weather, so you may want to bring an umbrella, but we still hope for some beautiful days. But that's all I want to say now, so see you there in 2 years' time.
Great, wonderful, and thank you very much, Gerhard. We look forward to it. So I guess that's it. We are wrapping up, and I would like to thank all of you for coming and making this meeting a success, which it was, and I'll see some of you tonight at the banquet and others maybe in Galway. Thank you.